Hello everyone, this is Dr.Priya Pillai, Assistant Professor from Clinical Nutrition and Dietetics, St. Teresa's College. Today we are going to learn about growth curve, which is also known as microbial growth phases. When we talk about microbial growth phases, it is very critical to understand because it forms the very basis. We have to play around the various stages to preserve food or to promote fermented foods. The pattern of microbial growth depends on internal environment of food, which is also known as intrinsic factor, as well as storage condition under which it is stored, which is also known as extrinsic factors. Since they may either support or discourage the growth of microorganisms. The growth of microbes in food or an artificial growth media follows a definite course. This pattern of microbial growth in a food or an artificial media, seeded, inoculated with any microorganism, can be studied in the form of a microbial growth curve. The growth curve can be obtained by plotting the count on the bacterial cells against the corresponding storage time under the condition favoring growth of microorganisms. A microbial growth curve would show the following four phases. As you can see in the diagram, the first phase is called as lag phase. Initially, there would not be any appreciable increase in the number of microbes, though there may be an increase in their size. This initial period is the time required for the adaptation of microbes to the new environment, which is food or media. During this period of microbial adaptation, adequate quantity of necessary enzymes and metabolic intermediates are built up inside the microbial cells, as these could be required for subsequent multiplication. So this phase is a beginning phase. Then we have the log phase, which is also known as logarithmic or exponential phase. Following the lag phase, the cell starts dividing by binary fission. Now, what is binary fission? That is, one cell is divided into two in each cycle. So, thereby doubling their number with each successive cycle of cell division. In this manner, the microbes increase in their number exponentially or by geometric progression with time, that is 1 to 2, 4 to 8, 16 to 32 and so on. Therefore, if the logarithm of the variable count is plotted against time, a straight line will be obtained. The next phase is called as stationary phase. After a varying period of exponential growth, cell division stops due to depletion of nutrients and accumulation of toxic products. The number of progeny cells formed is just enough to replace the number of cells that die. As an equilibrium exists between the dying cells and the newly formed cells, the viable count in these phases remains stationary. We have the last phase, which is known as phase of decline. This is the phase when microbial population decreases due to cell death. Besides nutritional exhaustion and toxic accumulation, Autolytic enzymes may also cause cell death. Thank you all for the patient listening.